afternoon. Thank you very much. I'm very happy to be at IASc. I hope there are some biologists in this audience because I noticed you're one. Thank you. Great. Okay. Few and far between, but outnumbered by the physicists and mathematicians, I think. So I'm here to tell you about something that has been bothering me for some time. And I work in nutrition, and this is something that the National Institute of Nutrition puts out as to what you should be eating in your plate every day. And you will see that there is half that plate is vegetables and fruits, and only a third is cereals or grains like rice and ragi and wheat and so on. 1/3. Most of it is something else. But if you actually look at the food people eat, it's all rice. You go to a school and you'll probably find they're eating brown rice one day, red rice another day, yellow rice another day. It's all rice. There's hardly any diversity in that diet. And to that extent, it's worrying because we keep thinking how many children are stunted in India, how many of them are not developing adequately. So the solution so far has been, well, if they're not getting the nutrients that come from a diverse diet, why not just put chemicals into their diet? For example, put supplements of vitamins into their diet, put some metals into their diet. So you get, adolescent girls getting pills that contain iron and folic acid. We get fortified salt, fortified rice. But nothing works because, you go back to Aristotle, who said the whole is greater than the sum of its parts, and that applies to the diet, that you can't really split it up into or disaggregate it into its nutrients, feed them all separately and hope that they would have the same effect of them being together in the diet. Now, the fact still remains that if you take a sentinel nutrient like, for example, vitamin B12, which I hope all of you know, vitamin B12 can only be found in sources that come from animals. You don't find them in plant foods. So animal foods are considered one of the diversifications. I know there may be some vegetarians who will disagree with me, but milk is, for example, an animal food. And so it's only vegans who don't eat vitamin B12. Now, if you look at the intakes of vitamin B12 in this country, they're terribly low based on international standards. So you'd expect, based on that terribly low vitamin B12, that 100% of us should be B12 deficient. But we did a national survey of Indian children, about 100,000 children, and we found a prevalence of B12. If you look at it here, at a max it went up to about 30% in some age groups in adolescence, but at younger age groups it was about 10%. And that was truly surprising for us. And here's the real kicker. You'd think that a lot of people would be clinically B12 deficient. Now, if you're clinically B12 deficient, you'll probably be anemic. You'll probably have pins and needles in your fingertips because you'll have some form of neuropathy. Nothing. Nobody had any clinical signs. So this was what puzzled us as to, okay, if you say we eat such little amounts of B12, why are we not so deficient? So the questions we began to ask ourselves was just for this sentinel nutrient. Remember, this occurs with proteins, it occurs with other vitamins and so on, but I'm going to focus on B12 for this talk. And could B12 absorption, for example, from that minuscule amount that you get from milk or eggs or whatever you may choose to eat, could that be upregulated so that you really absorbed it all very well? Or could you be such an efficient person that you did not excrete B12? You hung on to it and you recycled it. Or could it be something else? Whatever it was, we needed a tracer labeled B12 molecule to figure out all these things that was safe for human use. The reason I say safe is that when we looked at the B12 tracers that were available on the market, these tended to be radioactively labeled with either 14 carbon or 57 cobalt. And that's a big problem. You go to any medical school ethics committee and they'll say nothing doing, you cannot give radioactive things to human beings. Sorry, go away. So what we decided was, let's synthesize our own or biosynthesize it. So we got a culture of Salmonella enterica and we cultured it in our lab and we fed it with substrate called ethanolamine, which was labeled with carbon 13, which is a stable isotope, safe. And once we did that, we were able to purify cyanocobalamin, that was, or vitamin B12, that was produced by this salmonella. We confirmed it by mass spectrometry. We did a lot of animal safety studies because you're working with cyanide and you're creating a molecule that was linked to cyanocobalamin. And finally, we got ethical approval to give this to humans, and we were able to do human absorption studies and then began our answering of all the questions that we had raised before. So we started with an oral 13C labeled vitamin B12 dose, and we gave it to humans, and we followed the absorption in the blood of this, and we constructed a 2-compartment model of the kinetics of vitamin B12, labeled B12 appearing in the plasma. And the old labeled B12 with cobalt, radioactive cobalt, gave a test called the Schilling test, which is used in clinical medicine and is no longer used, except if you're really sick. But 70 years after Schilling first published his method to measure the absorption of B12, we then did it ourselves with 13-carbon cyanocobalamin, and we found a bioavailability of about 50%. And note here that you'll see that the absorption occurs at a peak at around 6 to 7 hours, and that's because the majority of B12 is absorbed from the last part of the small intestine, what we call the ileum. However, just for curiosity, we kept sampling the blood, and we found, to our surprise, that there was a second peak that appeared of the appearance of labeled cobalamin. And that surprised us because where's this coming from? Well, there are different ways. One possibility is what we call an enterohepatic circulation, where B12 is absorbed, as you can see, from the ileum. It goes to the liver through the portal circulation, and there it will appear in the plasma, where you can take a blood and you can measure it. But part of it gets re-secreted through the bile back into the intestine, in which case you would expect to see a second bump of a recirculating tracer. This is one of the problems that really bedevils a lot of the work we do with stable isotope tracers. So that could explain the bump, but the second explanation is a little easier. It is, well, if you could absorb vitamin B12 from the colon, that means it's gone past the ileum into the colon, then you'd get the same bump. So the question was, what do we do? And I was very fortunate to have a pediatric surgeon who's sitting here, Shalini, who she's one of my fellows, and she works a lot on several things around pediatrics. And she was very interested by this, and she suggested, why don't we just put some labeled B12 directly into the colon during a colonoscopy? I don't know if any of you know what a colonoscopy is, but it is where you put a tube up the backside of a person, all the way, as you can see, through the descending colon, through the transverse colon, all the way through the ascending colon, almost up to the cecum, which is where the ileum ends. And you can actually there, once you're there, you can squeeze in some B12, labeled B12. The point is that Shalini had to go to the colonoscopy lab, and there she had to talk to all the patients who are undergoing colonoscopy and get their consent to have this done, because the key was we needed to find healthy colons. So the gastroenterologist who's at the bottom here, Mallikarjun, would go all the way there, and if he said, yes, it's all good, clear, normal colon, we then inject the B12 into that person. If it was an abnormal colon, sorry, we weren't into measuring B12 absorption in abnormal colons. So this was done. Again, we constructed a compartmental model, and you can't keep a patient after having a tube up his backside to say, now sit with us, give us bloods. They're going to say no. They're going to say, well, 4 hours is the maximum, we'll stay with you. So for 4 hours, we continue to take bloods, and you'll see there in that graph, you can see how labeled B12 appeared in the blood, meaning it was being absorbed, and about 8% of a 5 microgram dose was absorbed. And that was fantastic for us because it showed that that could be a reason for that second bump. I'm not saying enterohepatic circulation doesn't occur. It could, but it looks like this might be a major reason. there was one problem we had, and we still have, which Shalini is actually solving for us. And that is that when you put a needle into the vein of a person, you're measuring the blood that got past the liver. But once the blood goes through the liver extracts a lot of nutrients out of it. We call this first pass absorption. And what appears in the plasma is what the liver doesn't want. So you really don't know how much was absorbed. So we need to figure that one out. So we're doing that, and Shalini is actually, you can do it by giving the same labeled B12, give it orally and IV. And if you can give it orally and IV, you can actually work out mathematically what is the first pass disappearance that occurs in the liver. This is an experiment that was done with riboflavin. So what we are doing is we have this really smart guy who came and joined us from IIT Madras, who's some kind of biotechnologist, and he is synthesizing, biosynthesizing two types of labeled B12 for us. One is an M+4 B12, and the other is an M+1. So on a mass spec, we can actually differentiate them. One will be given orally, and one will be given IV. And this was where we really began to see that, well, it looks like we're changing some dogma, because if you go to medical textbooks and try and look at it, basically the dogma is this, that once you're past the small intestine, that's it, you're dead. You're in a zone of excretion. Nothing gets absorbed there except water and maybe some salt and maybe some other small molecules, but not nutrients. But the fact is that we think that this dogma has to be reversed, and this is beginning to appear in textbooks from the papers we have written. And there are several bugs in the microbiome that can produce all those vitamins that you can see there. And for B12 particularly, in vitro, there are experiments that have shown that the human gut biome has the ability to produce up to 0.6 micrograms per ml of whatever brown stuff is in the colon. So it's a lot. So Shalini actually then applied to ICMR, which came out with this grant RFA, which was what they call the first in the world. It was to do research that no one else was doing in the world, do it for the first time. And so her point was, let's trace what these bugs are doing by offering them labeled substrates to ferment, and then see what nutrients they produce. And so she got this grant, the first in the world. They give you up to 15 crores to develop it's like you keep getting more and more money as you think of new things to do. So right now what she's done is she has introduced 13C sugars into the colon. Now if you put 13C colon orally, it'll never reach the colon, it'll be absorbed. But you can give 13C cellulose, and that reaches the colon, gets fermented, and she's looking at 13C B vitamins in the plasma and in fecal samples, which are not really the best thing, but they give you some index. So this is a sort of experiment she does. She gives 13C cellulose once, and then she kind of follows them for 3 days, taking blood samples every so often. And this is what she's found on something like 15 subjects, that there is a production of of B12 that appears in the plasma. So I think there's no doubt that if the cellulose reaches the colon, this can be produced, and you will get B12. And when you model it using first compartment kinetics, we find that you absorb about 1 microgram of B12 per 2 days. Just in context, your requirement is about a microgram per day. So you can get up to half of your requirement through this. And now we are actually looking at the microbiome and trying to look at the abundance of the microbial B12 synthesis genes. So you can see the pathways on that side, and we have mapped all those genes here. And so there is evidence that there's an abundance of genes, and then we have actually mapped those to specific bacteria. And you can see that these are the bacteria. This is just 9 subjects. We've got a long way to go. But my point is that we are on the way to actually figuring out which bug produced it and so on. And so I'm going to end here. So there are several necessary and several unnecessary questions with this. The necessary questions are, I mean, what else can the microbiome produce? So Shalini is actually looking at riboflavin, thiamine, pantothenic acid, folate, I mean, a number of B vitamins. We could look at vitamin C as well. The important thing is, with what precision can we measure the appearance of these vitamins in the plasma. How much of first pass did we miss? So we need to have two isotopes, IV and oral, which is what she's doing with the first. First in the world is not just B12, it's several vitamins. And more important, how can we measure functional outcomes? I think there are other nice questions. For example, what makes the altruistic bacterium? Because why on earth would a bacterium synthesize large amounts of vitamins and then secrete it? What told it to secrete it? What is the crosstalk? Is it a host-based thing? Is it a microbial ecology? These are really interesting questions that we look for in the future, but there are unnecessary questions that I get asked all the time. I hope you won't. It is, do we need to eat food at all? No, we do. We certainly do. There's a silly season for all science, but I hope we don't go there. Thank you.
I think you conveyed such a beautiful piece of work in such an amazing line. And lots of questions in my mind, but first the audience. I think Thelma and yeah.
There are all these probiotics that people keep recommending that you have. Is there a way of designing a good probiotic soup that you can introduce, for those who are deficient in B12?
Yeah, actually, the reason Shalini got, she's sitting there, by the way, the one in the yellow.
Oh.
The reason she got the first in the world was she said she would do this. She would design a probiotic for public use. Because our biggest problem is this, that we go and dump these chemicals into the food of people. Large amounts of iron, for example, which completely changes the biome. And to my mind, reduces its ability, perhaps, we have to check this, to synthesize these things because you get ferrophobic bacteria that hang out, or ferrophilic bacteria. So you're right. So our aim is not only to produce the probiotic, but a prebiotic. Figure out a mixture of the food that it likes, because there are different fibers. And we're thinking that one of the cheaper ones to give is a fiber called inulin. And we're trying to investigate that as well. So thank you. Yeah. You should get the first in the world prize.
Yeah, the other question which I had was that you talked about children to start with in your survey that you found. So do you assume that they really had a very healthy microbiome? And that is why you really kind of got such a high percentage of B12?
Absolutely right. Thank you for that. They are big assumptions because when you do these national surveys, it's kind of, you know what survey is going on for the caste system in Bangalore, where they just go and stick something on someone's door and say, we survey. You don't get much time. So we could not get stool samples, but there are many issues around B12. One issue that some very good investigators in Vellore had suggested was many people get their B12 from sewage. So you drink dirty water, but look, if you drink sewage, you will get B12. You may get many other things, but it was a suggestion at the time that could be one way. But now, as we have cleaned up our water, it's unlikely. The other is that maybe people eat more non-vegetarian food that they let on. Most people, if you ask them, they say, no, I don't eat any. And many people, when you go and ask them, do you eat well? They say, no, I don't, because they hope they're going to get a handout. So there are lots of psychological issues around this, but thank you. I can't answer your question, but I think they had a reasonable microbiome.
You have to evacuate the
Very good.
So
Very good.
Doesn't that potentially affect the results that
No.
that you get?
No. The reason is, the question we were asking was, does the mucosa absorb the labeled molecule? Not, does the microbiome produce that molecule? So when we did the colonic experiments, it was clearly only to show that there was a potential for absorption. Once that's done, what Shalini is doing is she's putting cellulose into a non-treated colon. You're absolutely right. If you look at the colon that is undergoing colonoscopy, it's brilliantly clean. But that's not the normal situation. So the only way to test whether there's fermentation is to have a non-cleaned colon, just give cellulose to just free living people and track whether there's this absorption.
One last question.
Thanks for fascinating information. I heard about 5-6 years ago that insulin is secreted by a group of bacteria. So the tablet metformin is actually a food for that group of bacteria. That was Nature article. You can- So
I wish I could answer that. Shalini, do you know anything? No.
I have the reference and so on. I will give.
That's
In fact,
fascinating.
I heard the talk by that Norwegian gentleman who came to Chennai and gave a talk on diabetic center. So I talked to him. So it's a Nature paper.
I'd love to see that because I don't know if there's any insulin absorption that could occur. I have to look at that.
So the bacteria produces insulin.
That's fine.
And the metformin is a food for that bacteria.
That's fine.
Yes.
How does the insulin get into the body is my
I do not
question.
know.
That's what I think I'd love to see that.
Sure.
Thank you.
Thank you so much. Just a small question that would the vitamin B12 levels need to be reassessed as to what would comprise a range for vitamin B12 because that's what you started your observation with, and would then getting a lot of people supplemented with vitamin B12 be not needed?
It's not a good thing. Please, for all of you who pop pills of multivitamins, not a good idea. Just eat a diverse diet. There are people who have died because they take too much of multivitamins. So please be careful. However, you're absolutely right. The diagnostic cutoff for B12 is a matter of great debate. Different countries have different levels. In India, we have not yet decided what that level should be. So we just follow what others take.
I think we do that for everything, for diabetes and all, and high time, somebody took it up. ICMR has been talking about it for 20 years, but there are no values in India. But
That's
because
true.
the diet of Indians are very different from European diet. And
We don't even know what
still
India
follow.
eats.
Yes.
That's the problem.
Yeah.
Somebody told me, some doctor, don't be too clean. That's the secret of many of them.
Absolutely. Give yourself some immunity, some resistance. Yeah.
Thank you
Thank
so
you.
much.
Thank you all.
Thank you.